Good afternoon, everyone, and welcome to MitoAction's monthly expert series. Today, Dr. Peter McGuire, Principal Investigator of the Metabolism, Infection, and Immunity, the mini-study, will join us to discuss Leigh syndrome, a factory for making viruses. My name is Stephanie Harry. I'm one of the patient support coordinators at MitoAction and will be your host for today. Today's presentation will be recorded and available on MitoAction's website in the coming days, as well as on our podcasts, Apple Podcasts, Google Play, and Spotify. We encourage you to ask questions throughout the presentation using the Q&A feature on the bottom menu bar of your screen. If you're calling in via phone, please feel free to submit your questions to us by email at info@mitoaction.org. We will do our best to get through as many questions as possible at the end of today's presentation. Previously, the McGuire group at NIH reported that children with mitochondrial disease in general experience serious infections which may last longer than expected. Cells impacted by mitochondrial disease present a unique environment that may be facilitative for viral replication. This relationship between compromised mitochondrial function and increased viral activity suggests a complex interplay where cellular energy deficits might inadvertently support the viral life cycle. Dr. Peter McGuire received his bachelor's in psychology from Villanova University at master's in microbiology and immunology from New York Medical College and a MBBCH with honors equivalent to an MD from Royal College of Surgeons in Ireland. After completing a combined residency in pediatrics and medical genetics at Mount Sinai Medical Center, he was awarded a fellowship in biochemical genetics from the American College of Medical Genetics. After completing his training, he remained as a junior faculty member in the program for inherited metabolic disease at Mount Sinai. Dr. McGuire is board certified in pediatrics, clinical genetics, and biochemical genetics. In 2010, Dr. McGuire moved to the National Institute of Health, the NIH, to join the Physician Scientist Development Program to accelerate his translational research program. He was appointed to the position of tenure track investigator in 2016. Throughout his career, Dr. McGuire has focused on improving the care of patients and disorders of mitochondrial metabolism. By combining his training in immunology and biochemical genetics, he fashioned a translational research program to understand host-pathogen interactions and disorders of mitochondrial metabolism. His NIH Clinical Center Protocols, the NIH mini-study, is the first organized effort to study viral infection, immune function, and disease progression in patients with disorders of mitochondrial metabolism. Dr. McGuire, we love you. What can I say? I mean, you're amazing, and you are really helpful in helping us to better understand infection, immunity, and just breaking everything down. We really appreciate you coming on today, sharing more about your research, and just continuing to educate us about viral infection.
Thank you so much, Stephanie, for that introduction. And I just wanted to say Happy New Year to the community out there. I know maybe it's a little bit late. It's already February, but I still wish people a Happy New Year. Thank you to all the families who have participated in our study. And of course, we always are welcoming new families to come join us so we can learn more about infections in these children and how we may help mitigate some of the effects. So let me share my slides. Okay. And that looks good.
Looks good.
Okay, great. So I'll get started. So my talk is a little bit, I changed the title just a little bit, just to focus on influenza, because that's a lot of the work that we do with influenza. Although this does apply, by the way, not only to influenza, but to other viruses, and also applies to other types of mitochondrial disease, not just Leigh syndrome. So the purpose of my section here at NIH, the metabolism, infection, and immunity section, is to help define the risk factors and mechanisms that are involved in infection-related clinical decline in children with mitochondrial disease. And we do that by conducting clinical research. So once again, thank you to all the families who have come to visit us. From those observations that we make at the NIH Clinical Center, we then come up with these hypotheses or basic questions that we want to test. And we do that using model systems. And that can sometimes be blood or tissue from patients, or sometimes that can be animal models. Today, I'm going to tell you about an animal model that we use. And the idea is by understanding these mechanisms, we can eventually develop treatments which we can bring back to the clinic or even make recommendations to our patients or to the mitochondrial disease community as a whole. So that's our goal. So this is my study, which is the mini study, which is the metabolism, infection, and immunity study. And you can participate by coming here to the NIH, and we provide, we cover all the travel and things like that to have you come here. We also have individuals who participate remotely, so you can participate at home as well. So please feel free to contact us if you have any questions about how you can participate in our study. It's a natural history study of infection and immunity, particularly viral infection in kids with mitochondrial disease. And we study things like the infection history, since the immune system is very important for fighting off infection. We study the children's immune function. And then we also study the disease itself and any disabilities that may result from the disease, as well as from any infections that they may have, because viral infection can actually cause mitochondrial disease to progress. Okay, so what do we know about viral infection and mitochondrial disease? So first of all, up to 80% of children with mitochondrial disease experience recurrent infections. So in other words, repeated infections. And some of these can be quite severe and require high levels of hospitalization, sometimes even the ICU. And most of these infections are actually respiratory infections. So these are viral infections, actually, that the kids are experiencing. They can cause something known as, which clinicians cause metabolic decompensation, or kind of a deterioration in metabolic function, in this particular case, mitochondrial function. But it does occur in other types of inborn errors of metabolism as well. And then infection itself is the leading cause of neurodegeneration in episodic neurodegeneration in mitochondrial disease. And so I'm showing this to you here on the right in the lower corner, where this is actually an MRI of an individual who had an infection and actually had deterioration of their neurologic disease, and this individual had Leigh syndrome. And the red arrows are kind of pointing out in panel B, this is actually the brainstem down here where the red arrow is pointing, and you can see these kind of areas of light white. I'm not sure how well it translates on the screen, but you could just take my word for it that there are kind of these areas of white in here, and this is swelling in the brainstem, and that actually is as a result of neuronal death. So following this infection, this individual developed neuronal death inside their brainstem, which I'm also showing you here in panel C. I know I saw someone raise their hand to have a question. I will answer all questions at the end, if that's okay. Okay. So I encourage you to maybe write down your questions as you're watching, and I'd be happy to answer them. So with infection, as I mentioned, viral infection is a leading cause of metabolic decompensation in patients, and it causes life-threatening bioenergetic failure or even organ failure, because a lot of different organs require a lot of energy to be able to function. And so if we look here on the right, you can see that children, as they develop certain skills, which are part of their normal development. Kids with mitochondrial disease actually will show a stepwise decline in their skills. And this can happen as a result of the progression of their disease normally, but it also results when they actually have a viral infection, which then exacerbates their disease. So they can actually lose milestones, which they will never regain. So what we can see is kind of a stepwise regression in their development. So kids with mitochondrial disease, besides having mitochondrial disease itself, which can be a risk factor for having adverse outcomes as a result of infection, they also have other things which also can make them susceptible. When we did our study about 2 years ago during COVID, to look to see, what kind of comorbidities did kids with mitochondrial disease have, which could put them at risk for adverse outcomes during COVID or any viral infection for that matter. They had things like asthma, which is a risk factor for adverse outcomes with regards to viral infections. They also have, things like problems with swallowing and breathing, immune issues where they either have a diagnosis of immunodeficiency or demonstrated immune dysfunction. Um, Also, things like hygiene, hospitalization, even though we do have to go to the hospital, sometimes to help us with, or for patients to help them with their mitochondrial disease during episodes of infection or things, or even, medication issues or seizures or things like that. That also is an infection risk, because unfortunately there are a lot of people in the hospital who have infections and carry infections. Also, any types of devices like access devices, like a central line or something like that, which an individual may have for some reason who has mitochondrial disease. These also can be sources of infection. So there are other things besides having mitochondrial disease which can make an individual susceptible to infection. And these are things that we have to think about when we have a multidisciplinary approach to, treating patients as well as advising them about the risks of infection. So what types of viruses do kids with mitochondrial disease get infected with? Well, it's all different types, including influenza, influenza A and B. But I'm just showing you here some of our data from our study that we did, where we monitored for 1 year's time individuals during the COVID-19 pandemic. And so the kids that we monitored, this was actually like 20, it's actually families. So it's not just kids with mitochondrial disease, but it's the entire family. Because the family can obviously, as we all know, we can all share viruses in our households, and that's a risk to the child with mitochondrial disease. So this is the hit list essentially of what we saw in 1 year's time during COVID. So you see things like enterovirus B, which can cause kind of GI symptoms and things like diarrhea, as well as enterovirus C. But then you see a lot of respiratory viruses like rhinovirus A and B, which causes the common cold. You see influenza B, respiratory syncytial virus or RSV, which is a big one that's being kind of talked about now, as well as mass adenovirus, and then of course coronavirus as well. And so what was interesting is one of the most commonly shared viruses amongst family members as well as the child who had mitochondrial disease was enterovirus C. So anyway, so these are studies that we do in the home as part of participation in the mini study where we send kits out to individuals, and we continue to monitor families over time to see what viruses the individuals have been exposed to. And the reason is because we've actually realized that not every viral illness causes decline in our patients. So we're starting to ask questions about, are there particular viruses which are worse than others? So that's one of the reasons we continue to monitor our patients who participate in our home study. So what is a virus, essentially? So they are submicroscopic, so very, very small infectious agents, and they infect all life forms. So plants have viruses which infect them. Even bacteria have viruses which infect them. So viruses are some of the most abundant infectious particles in the world and can survive in all sorts of environments. But the thing about a virus is it's not actually considered living because it needs a host to replicate. So a living cell, viruses have to infect living cells, whether that's an animal cell or, like a skin cell or a cell lining your respiratory tract or a plant cell. They all need the mechanisms inside those cells to help them replicate and make more viruses. And they contain a core material, which is nucleic acid, which is either DNA or RNA. So how are viruses transmitted? And this is part of, sometimes some of the behaviors that physicians will advise. Some of the things that are advised during COVID-19, which kind of are targeting the way that viruses are particularly transmitted. So things like touch or respiratory droplets, sometimes direct contact like EBV used to be called the kissing virus, or which is mononucleosis. Also, there's obviously blood transmission for things like HIV, but also contaminated water, noroviruses, or, you always hear the stories about cruise ships where they have an outbreak of a diarrheal disease, and those are usually viruses that cause that. which can be transmitted in populations that are very close together. Insects like Zika virus, which we heard about a couple of years ago. And then even during childbirth, so cytomegalovirus is actually a virus that actually can cause birth defects and calcifications in the brain and all sorts of problems. So viruses can be transmitted in many, many different ways. The ones we're going to talk about today, or the one we're going to talk about today, is a respiratory virus, influenza. So there are many different factors that affect the transmission of viruses. There are seasonal factors, such as, the temperature, the humidity, the amount of sunlight. Sunlight actually is able to destroy viruses or actually break down viruses. Indoor factors like the temperature, humidity, as well as the air exchange within the environment that you're in. Host defenses obviously are really important. We have barrier defenses, lining our respiratory tract that can actually help protect us against viruses. And that includes things like, mucus, as well as, the barrier of the cells themselves. The cells have antiviral defense mechanisms that they can use. Also, the clearance of mucus and things like that, which is called the mucociliary clearance. These also can affect host defense. The virus itself, factors that affect its transmission is how viable the virus is in different scenarios. So you probably heard during COVID that, SARS-CoV-2 survived on surfaces for a certain amount of time and things like that. And so therefore you should clean surfaces. The dynamics of the droplets themselves. So respiratory viruses can be transmitted by these these microparticles of fluid, essentially, which then can penetrate into respiratory passages. And then how, the virus is actually transmitted, whether it's airborne by aerosols or droplet sprays. Those are kind of size differences, or even by indirect measures, which can then influence transmission between an infected host and a susceptible host. But also are the things that we can modify, like our human behaviors. So for instance, whether we're in school at settings or things like wearing a mask, social distancing, some of the things that were recommended during COVID, as well as hygiene practices. Hand washing is probably, and not sharing utensils and things like that, is one of the strongest methods for preventing transmission between individuals. So there's a seasonality, as you would imagine, to different viruses. And right now we are in the middle of flu season, but it's also RSV and also COVID season. Today, I'm going to talk specifically about influenza. So this is from the CDC's website. So you can go on the CDC website and look under flu or flu. It's actually called Flu View. So it's a nice little catchy phrase there, which highlights the number of influenza cases in the US for flu season. So you can see here at the bottom, it goes from 2023 into 2024. We're now, and so it's 2024-02. That means we're in the second week, or this goes up to the second week and beyond of 2024. So there's a little bit of a lag, right, of collection of this information. And this is influenza positive tests in US clinical laboratories. And you can see during around the November-December time for this year, we had a spike in number of positive cases by laboratory detection. So these are hospitals that are reporting this, clinics that are reporting this through laboratory testing. So a spike has kind of occurred around November-December time, and now is starting to decline. This spike shifts every year. So it sometimes can remain the same around November, December, but sometimes can shift a little bit later. And during COVID, we had a little bit of a shift of timing of viral infection, particularly influenza infection. So what types of influenza is actually occurring? So during this time, during the peak, as well as actually during the whole time where we've been monitoring influenza for this influenza season, which begins at about week 40, which I think is around November or so, or October, and goes through to about March or so. And so most of the types of influenza that we're seeing are H1N1. And this is a descendant of pandemic 2009 H1N1. So if everybody remembers back in 2009, the H1N1 pandemic, this virus is still around. So it's an influenza A subtype. Um, Other types that you can see are H3N2 and influenza B virus. So there are different subtypes of viruses. The way that some of these viruses are named has to do with the types of proteins they have on the surface. So the H stands for hemagglutinin, N stands for neuraminidase. And so this is H3 or hemagglutinin type 3, neuraminidase type 2. Same thing for H1N1. It's type 1, type 1. So this is kind of one of the ways we name viruses. Okay, so this is actually the percent of outpatient visits for an influenza-like illness during this current flu season. And it's actually divided by age group. And so the age group that shows some of the highest amounts of visits to outpatient facilities is actually the 0 to 4 age group, so children. So, Leigh syndrome falls into this age group, so they are, at risk for, for influenza-like illnesses. Across the country, there are some differences in the number of cases. So this is for the week 3 of the 2024. So it would be 2024-03. Basically, looking at influenza cases, reported influenza surveillance across the United States. And you can see that the darker it is, the higher the number of cases. And in the Southwest, there seems to be in January, a large number of influenza cases, where up north there seems to be less. This will change over time, of course, because influenza is highly infectious and spreads actually quite easily. California also, as you can see, has a good number of cases as well. So a lot of states are in kind of the high category, even some in the very high category. And so this is the reported number of, so influenza, besides causing kind of the flu and feeling kind of crummy and having muscle aches and things like that, there is a mortality. Before COVID even came along, influenza does have a significant mortality that's associated with it. So this is actually the number of pediatric deaths from 2020, the 2021 season up until the current season now. And last year, for influenza-like illnesses, 2022 to 2023, there were 183 pediatric deaths. So this is just children. In 2024, we're already up to 57. So it's not, influenza is not kind of, even for individuals who don't have mitochondrial disease, it's not an insignificant thing. I mean, there is a more significant morbidity that is associated, but also significant mortality. So there are a lot of ways, to help prevent the spread of flu, and the CDC has all these really nice, colorful posters that encourages things like rest and staying away from other people if you are sick, covering your coughs and sneezes, as well as hand washing. Of course, get yourself vaccinated. And then if you do get diagnosed with flu, there are some medications that you can actually take, antivirals such as Tamiflu, which I'll talk about a little bit later. So influenza itself is, it's a type of virus. As I mentioned, viruses have nucleic acid inside them, and that's either DNA or RNA. And in this particular case, influenza is an RNA virus. And as I mentioned to you before, they're actually typed by their surface proteins. They're hemagglutinin and they're neuraminidase. So they can be called H1N1 or H3N2. They still remain a major public health burden and have the capacity to cause pandemics. They actually can lead to systemic inflammation, which can lead to mitochondrial disease exacerbation. This is from a paper we published years ago, which was a mitochondrial disease model of liver disease, so mitochondrial disease in the liver. And that liver disease got worse when we gave that mouse influenza. So it can exacerbate mitochondrial disease symptoms. It could also exacerbate central nervous system disease in a mouse model of Leigh syndrome, which is what we're looking at now. So this is data we haven't published this yet, but we have noticed that when we infect this mouse model of Leigh syndrome, The mouse normally has seizures. Those seizures get worse, a lot worse, and the mice can even die when you give them influenza. So influenza can be pretty serious in mitochondrial disease. And so this is what the influenza virus looks like. Here on the left, this is kind of a cartoon version. And on the surface, you can see the red spikes are the hemagglutinin. So the hemagglutinin is different types. So that's where the H1 comes from. And the neuraminidase is another type of protein. This is in blue, which is on the surface as well. And that also has different types. So H1N1 is type 1 hemagglutinin and type 1 neuraminidase. This is what the virus actually looks like under a microscope. And you can see it is circular. It looks kind of flat because this is not in 3D. But here on the bottom, you can see what this is this is a cell that's infected with influenza. And you can see the little viruses actually budding off from the cell. So this is what happens in our respiratory tract, is that cells lining our respiratory tract, including our upper respiratory tract as well as our lungs, can get infected with influenza. It replicates inside the cell, and then it buds off to infect other cells. Okay, so this is what the life cycle of influenza actually looks like on the level of the cell. So these cells lining our respiratory tract can actually get bound to these sialic acid-covered receptors on the surface. So proteins on the surface of a cell are like decorated with sialic acid residues. And these are kind of like sugar, essentially like sugar tails. So they're sugar tails that are on proteins and on fats that are in the membrane of cells. So that's the target that influenza actually binds to by hemagglutinin. Hemagglutinin binds to sialylated, that's a mouthful, but sialic acid covered receptors or proteins on the cell surface. Hemagglutinin binds this, and then it enters the cell by endocytosis. Once it goes inside the cell, the RNA that is part of the genome of the virus gets released. It goes to the nucleus, and it does two things. It replicates itself, but then it also makes a bunch of proteins that are important for making more virus. Things like M1 and P protein and pol protein. It's not important to know the details, but just to demonstrate that the virus makes a number of proteins itself using the host's mechanisms to make those proteins. So it hijacks essentially the mechanisms inside the cell to make these proteins. These eventually get placed on the surface of the cell, and all of these nucleic acids then get repackaged. The RNA gets repackaged inside this budding off the membrane here, which contains, once again, hemagglutinin and neuraminidase and some other proteins, and forms a fully formed virus. So this virus, influenza, like other viruses, needs a host to facilitate its replication. Otherwise, the viruses can't survive. So we want to ask this question, so do kids with mitochondrial disease, and we study Leigh syndrome, but we've also looked at other types of mitochondrial disease in children, do they get sicker during viral infection? So these are some data that we published a couple of years ago. It's two separate papers, but I included the figures from them. And so kids with mitochondrial disease get a lot of recurrent and severe infections just in general. And those infections are upper respiratory and lower respiratory tract infections. And most of them are viruses, essentially. And what's interesting is that in the second bar graph I'm showing you here on the right, the white is actually the number of positive individuals. And it turns out that kids with mitochondrial disease, their caregivers report in 80% of them, that 80% of the kids experience delayed recovery from infection. So in other words, their infections seem to last a lot longer than what they've noticed from themselves or other children or siblings or whatever. The kids just seem to take a while to recover. So what's potentially the reasons for that? So since obviously we can't invite people to the NIH Clinical Center and give them influenza and see what happens. Like that would be unethical to do. And it's one of the things we're trying to prevent. So we have to use mouse models. So there's a really good mouse model for Leigh syndrome, which is known as the NGUFS4 knockout mouse. So NGUFS4, just very quickly, it's just a piece of complex 1. It's one of the components of complex 1, which is important for extracting energy from your food. So eventually this goes on to make this transfers electrons down the electron transport chain and allows us to make ATP, which then allows our cells to do work. So that's the energy part, is the ATP that's being made from our food. So anyway, these individuals are they're also individuals who have NGFS4 dysfunction or mutations in genes involved in complex 1, especially NGFS4. And so these individuals have complex 1 dysfunction. So the mouse itself really replicates Leigh syndrome actually quite well. So the mice only live to about 60 days old or so. And these are homozygotes. So this is an autosomal recessive disease. So it means they have to have 2 bad copies of NGFS4 to display that complex 1 deficiency. They lose their hair. So they have this kind of premature aging that seems to happen. And as they mentioned, they die early at about 60 days or so. And these are some of the things that we can actually observe, like cachexia means weight loss. So by about, day 30 or so, they really start losing weight. And then they display other behaviors like ataxia because they do develop lesions in their brain. In Leigh syndrome, for those of you who are not familiar with Leigh syndrome, they develop neuronal death in their basal ganglia as well as their brainstem, as I mentioned earlier. And so they can have abnormal movements. And that's what ataxia means. They are kind of stumbling a little bit. They almost look drunk, as if they've consumed alcohol. And then, of course, their death, nearly 100% of them, or 100% of the animals die by day 75. So the lesions that are developed in Leigh syndrome, the kind of time course, a lot of things replicate Leigh syndrome. So it's a really good model. So we infect them with flu. So this is a mouse-adapted influenza virus that we use. And you can see here, this is the NGFS4 knockout mouse on the left here with the red writing. And you can see the hair loss here, right? They turn gray and they lose their hair. They're also a lot skinnier than the wild type mouse. So we place the animals in these baskets. We put them in this large infection machine, and we give them influenza virus, which is delivered by a nebulizer. So this is kind of like what the nebulizer does is it turns the influenza into really, really small particles, which then penetrate deep into the lungs. So we're essentially giving these animals influenza pneumonia. Okay, so the animals are infected, and then we collect samples at a couple of different days and time points. The other thing about this particular type of influenza is the mice actually can survive this infection. There are other types of influenza viruses you can use in mice where the mice die, but this is one where the mice actually live. So we can monitor them out to, or the wild type mice are supposed to live. So we can monitor them out to 10 days at least, and take different samples, looking at the amount of virus they have, taking blood samples, things like that. Okay, so mice that are infected, NGFS4 knockout mice, Leigh syndrome mice, exhibit increased weight loss when they get infected. So this graph here on the left is actually looking at the change in weight. And the good thing about weight, when you're doing any experiments in animals, is one of the best ways to tell if a mouse is sick is actually weight loss. Because when a mouse feels sick, it won't eat and tends to lose weight. So it's actually the best measure that we have, whether the mice get sicker or not. And you can see here, the red lines indicate kind of the weight loss in the mice. And so they lose more weight than the wild type mice. They eventually do gain it back, as the wild type mice do as well. But they really dip down more than a 20% loss, essentially, in their body weight. So they're really, really pretty sick. And so when we asked about, well, why were they getting so sick? The reason is because they have more virus in their lungs. So we looked at the amount of virus. This is known as the viral load in the lungs of these animals. And we looked at days 1, day 4, day 7, and day 10. And you can see at day 4, when they really have this, really one of the time points where they have a lot of weight loss, the amount of virus in their lungs is like almost twice as much. And what's even more interesting, going along with what we heard from families, is that my child takes a longer time to recover from infection and things like that. At day 7, when the amount of virus was undetectable in wild-type mice, it was still detectable in the Leigh syndrome NDFS4 knockout mice. So the virus actually persists for longer in these mice as well. So not only do they have higher amounts, it persists for a longer amount of time. And so so what happens as far as the immune system? So when the immune system gets activated, it's supposed to make all of these different chemicals called cytokines. So cytokines are made by immune cells, and they are made because they are important for fighting off any type of infection, whether it's a virus or a bacteria or whatever. The problem is that too much production of cytokines can make you feel sick, right? Makes you feel kind of ill, under the weather, gives you muscle aches, things like that. So when we looked at the ratio of how much cytokines are produced by the knockout mouse, the Leigh syndrome mouse, versus the wild type mouse, it turns out that the knockout mouse or the Leigh syndrome mouse was making much more cytokines. So in other words, there was much more inflammation in these mice, and that's why that's one of the reasons they were sicker. So they have more virus, which is stimulating a greater immune response where they make more cytokines, which make the mice feel sicker. And so we saw things like, interferon gamma, IL-6. The names are not important. The important part is that these are inflammatory cytokines that make you feel miserable. They're necessary for fighting off the infection, but you feel miserable. So yes, the Leigh syndrome knockout mice do become sicker during viral infection, as marked by weight loss and this increased inflammatory response. So why do they have more virus? What is it about having a mitochondrial, not just Leigh syndrome, but this actually applies, as I said, to other mitochondrial diseases as well. Why do they have more viral load? Why is there more virus in their lungs? So when viruses actually infect cells, so for instance, in this case, influenza, there is enhanced glycolysis, right? So in other words, burning sugar. So bringing sugar into the cell and using sugar as an energy source is actually enhanced. And the reason is because this is optimal for viral replication. So in other words, when it infects the cell, a respiratory epithelial cell, influenza, it then causes that cell to shift its metabolism towards glycolysis instead of mitochondrial metabolism like oxidative phosphorylation. So glycolysis shoots up essentially. And the reason this is important is because glycolysis is very biosynthetic. It helps the virus make nucleotides, which are important for making more viral RNA. It's important for making fats, which is important for making the viral coat, which is a lipid membrane. So basically, glycolysis is very biosynthetic, and the virus takes advantage of that by forcing the cell to make more, to burn more glucose, and that it can then use to make more virus. So we know that cells affected by mitochondrial disease, in this particular case, cells, so these are mouse respiratory epithelial cells that are actually knocked out for NDUFS4. So they have complex 1 deficiency, therefore mitochondrial disease. You can see that the amount of glucose that they use or burn, this is by something called ECAR, which stands for extracellular acidification rate. The point is that it measures how much glucose is being used. These cells that are deficient actually burn a lot more glucose, as you can see here. So these cells at baseline, in the absence of viral infection at all, before viral infection even occurs, glycolysis or the use of glucose is upregulated in these cells. So therefore, is this environment kind of facilitative for viral infection because these things are occurring already. And so if we could block glycolysis or kind of you can't block it completely, but you can dampen it down a little bit, can we actually limit viral replication? So to do that, we use a drug called DCA, which is known as dichloroacetate. And so this is a drug that actually inhibits an inhibitor. So that's kind of a mouthful and a way to a mechanism that you have to try and wrap your head around. But basically, this kinase here inhibits something called pyruvate dehydrogenase complex. Pyruvate dehydrogenase complex, when it is not inhibited, allows glucose to go into the TCA cycle and eventually get burned by the mitochondria for energy. When glycolysis is upregulated, this spout is shut off. So it's shut off by pyruvate dehydrogenase kinase. So glycolysis goes up, pyruvate dehydrogenase kinase becomes active, and it shuts off pyruvate dehydrogenase complex. So pyruvate, the end product of glycolysis, does not enter the mitochondria, does not get burned for energy. You're only then making ATP by glycolysis. So it shuts off the spout at the top where it doesn't flow into the mitochondria. So therefore, if we could block that by using DCA, so inhibiting the inhibitor, and allow glucose to flow into the mitochondria, or pyruvate to flow into the mitochondria, could we steal away these carbons that the virus is using from glycolysis to make more virus? So by stealing away from glycolysis, we could prevent viral infection. And so that's what we're trying to do. And actually, we were able to do that. So these are Leigh syndrome mice that are infected with influenza, where we treated them with DCA, and then we looked at the viral load in the lungs of these animals. And you can see dichloroacetate, by stealing away carbons from glycolysis and allowing it to flow into the TCA cycle, you're stealing away carbons from that virus for it to be able to replicate. And in doing so, we actually squash the virus. That's the blue bar here. We squash the amount of virus that's actually in the knockout animals that are treated with DCA. The other thing too is you squash a lot of inflammatory cytokines. So the inflammation gets squashed as well. So here I'm showing you two inflammatory cytokines, interferon gamma and IP-10, which also get downregulated when you give dichloroacetate. So decreasing the amount of virus, decreasing the inflammatory response. So just a quick summary. So in general, we have increased viral load in mitochondrial disease in the lungs of individuals. It causes cytokine storm, which could cause metabolic decompensation. And we can alter this process by altering glycolysis. But how does increased glycolysis actually help with increased viral load? In other words, is it just that it helps, it gives the virus, it produces more carbons that the virus can use to make more virus, or is there something else involved as well? So, to do this, we actually look in cells, and these are mouse respiratory epithelial cells that we infect with virus. So, we're kind of simulating essentially the cells that are lining the respiratory tract and how they get infected with the virus. And when we do that, when we infect those cells, you can see that the knockout, these are NDUFS4 knockout lung epithelial cells, they have much more virus, just like we see in the mouse. What's interesting is that also too, once again, when we treat them with dichloroacetate, we can actually decrease the amount of viral load in those cells. Also, if we treat with 2DG, 2DG also blocks glycolysis. So DCA steals from glycolysis, 2DG blocks glycolysis. Point being, in both scenarios, glycolysis is downregulated, both of which we see a decrease in the amount of virus in NDUFS4 knockout cells. So the treatment also works, and glycolysis seems to be upregulated, contributing to viral replication in these cells as well. So what's going on here? Why is that happening? How is glycolysis contributing to this? Well, one thing that we just first need to understand is the viral life cycle, which I went through before. And the part I'm going to talk about exclusively is attachment. So as I mentioned before, the way that viruses enter and bind is they actually bind to sialic acid residues, which are on proteins on the surface of the cell. So as you can see here, in blue is the hemagglutinin protein, and it's binding to the light blue sialic acid residue here, which is on the surface of a glycoprotein, which is on the surface of the cell, and it binds to, and that's how it binds, and it essentially enters the cell after making that binding. So the interesting thing is that influenza binding and attachment to the cell is very, very fast. It happens in about 10 minutes time. The entire viral life cycle itself to making from start to finish, making brand new virus after it enters the cell takes 8 hours. So we can look essentially at very early time points to look at this binding. And so when we treat cells with very, very small amounts of virus at this 10-minute and 20-minute time point, so before it even enters the cell, before it even gets to the nucleus, before it even starts to replicate, we see that there are lots of virus essentially stuck to cells affected by mitochondrial disease. So the MDUFS4 knockout cells have more virus actually sticking to it. So that is interesting. And so we asked, well, why is that so? Do they have more sialic acid receptors or residues on their surface for viral binding? So viral attachment happens by the binding to these, as I mentioned, sialic acid residues. And so there are particular ones, which the details aren't important, but there are particular types. And so we asked whether there was more sialic acid residues on the surface of cells affected by mitochondrial disease. And we do that using something called a lectin, basically something that just binds to sialic acid residues and allows us to detect how much sialic acid is there on the surface, how many binding sites are there for potentially for influenza. And it turns out that in mitochondrial disease, they have more. There is more sialic acid residues on the surface of mitochondrial disease cells. And so where is that actually coming from? Well, it turns out that the sialic acid synthesis pathway, the biosynthesis pathway, comes from glycolysis. So if we treat the knockout cells, the cells that are affected by mitochondrial disease, with blockers of glycolysis, whether it's 2-DG, which blocks glycolysis entirely, or it's DCA, which steals carbons away from glycolysis, you can see in the knockout cells, we actually are able to decrease the amount of sialic acid. And then I'm not showing this in this figure, but we also are able to decrease the amount of virus that is bound to the cells and actually gets into the cells. So the reason that kids with mitochondrial disease may remain sick for longer is they get a higher dose of the virus because there's more sialic acid residues on the surface, which are free to be bound by influenza virus. So they kind of get a higher dose of influenza virus. Okay, so there's enhanced essentially binding of this virus by the sialic acid residue, which allows more virus to enter the cell and replicate. So therefore, our patients get sicker. Okay, so the last thing I just want to mention has to do with, what drugs do we actually have to attack influenza or try and prevent influenza infection? And so the drugs that are either available now or are currently being tested basically attack the different parts of the viral life cycle. So this is just a different figure of the viral life cycle, which shows attachment, the bringing of the virus by endocytosis, the release of the viral RNA, it goes to the nucleus, all the things we talked about before, the synthesis, et cetera. So there are different drugs which are listed in red, which are basically targeting these different parts of the pathway. The one that we are most familiar with, and the one that's readily available, is Tamiflu. And Tamiflu is a neuraminidase inhibitor. And so what Tamiflu does is it prevents the release of virus. So the cells can still get infected. It's just that it doesn't get released to then go and infect other cells. And that's a process that actually multiplies the viral production. So Tamiflu actually blocks the neuraminidase, which is important for the release of virus and therefore prevents the spread of virus. So it doesn't prevent flu in the first place, but it may limit the length of time that an individual will be sick with flu because it's preventing spread. And so this is Tamiflu for some of you who have seen it before. It does come in a capsule form, and it works against both influenza A and influenza B. And it's recommended for individuals 2 weeks of age to 65 and above. There is low resistance. So as we all hear, the virus mutates, right? It makes changes in its genetic code to try and every year, which allows it to evade the immune system. But Tamiflu is actually quite good. It's not affected by this or less affected by this mutation rate, where it still remains effective even in viruses that have kind of changed. And then the most important thing is the time period. So basically 2 days, the 48-hour time period is very important for getting Tamiflu because it's most effective when it's started within the first 2 days of flu symptom onset. So it's important to go and get tested and talk to your pediatrician about Tamiflu. Okay, so in summary, influenza still remains a major threat to children with mitochondrial disease, a significant cause of morbidity and mortality in that population, even in individuals who don't have mitochondrial disease. Our mouse model of Leigh syndrome gets sicker during influenza infection, and the reason is because the lungs from those mice have higher viral loads, which stimulates the immune system very strongly. They get something called cytokine storm, really heavy inflammation. They feel miserable. They lose weight. The respiratory epithelial cells in the mouse that are affected by mitochondrial disease may actually be stickier. I use the word stickier. What I mean is that they have more sialic acid receptors or residues on their surface, which is recognized by influenza virus. So therefore, they get kind of a higher dose of influenza because there are more receptors to stick to. And then last but not least, which is the most important point clinically and kind of a clinical pearl, is that Tamiflu can help interrupt this viral life cycle, right, by preventing spread because it doesn't allow the virus to kind of exit and go infect other cells. So it's important that if you feel your child or yourself have flu, that you should contact your provider. And it's important for family members as well, because as I talked about earlier about the spread of viruses within families, Tamiflu within the first 48 hours is the most effective. So there are a lot of different people to thank. Of course, thanking Mito Action for inviting me here today. I really appreciate being able to connect with the community and with families, as well as all the providers who have, sent patients to us. And then the work I showed you today was the work of a really talented postdoc. Her name's Amanda Fuchs. And she did a lot of the mouse work, as well as Jillian Jetmore, who is a student in my lab, who's going off to medical school, who did a lot of the cell work with the sialic acid residues. And these are some of our other collaborators. So I'd be happy to take questions.
Thank you so much, Dr. McGuire. I love it. I always learn so much when I listen to your presentations. It's just about how the biochemistry, how things work. And we have a lot of questions filtering in. One of the first ones that came in that's not actually on the Q&A that I wanted to make sure we got to. If you decrease the glycolysis using the DCA, what are the mice using for energy?
Yeah, they're still using, so first of all, OXPHOS is never zero, right? Even in mitochondrial disease. OXPHOS will change, even in normal cells or cells not affected by mitochondrial disease, I should say, where it's kind of turned up and turned down, but the engine's never turned off, right? So the mice are still, able to use from glycolysis, even though we're tuning it down a bit. They still are able to get energy from there. They are still able to get energy from the mitochondria, even though it's tuned down a little bit, from fatty acid oxidation, which also feeds into the mitochondria. Doesn't work the best, but it still works somewhat, right? So they're still getting some energy from that. So it's not the most efficient system. So the mice, so when we block glycolysis, and they do seem to decrease their viral load and decrease the cytokines, et cetera, we still are robbing them a little bit of their energy because we're not feeding them more like we would normally do, right? Because you want to do, caloric supplementation and things like that during illness. So they still continue to lose weight because we're not addressing that specifically. So they can still get energy from their kind of what they are eating, but the idea would also be with any infection is to give more calories, right? Because your basal metabolic rate with infection can go up anywhere between 10 to 15% for every 1 degree of temperature. So caloric supplementation is going to be very important for that. In the mice, we didn't do that, so they did get a little bit sicker in the sense that, but in humans, obviously, it's very different because we would be hitting multiple things at once.
So then if you have a patient with a fatty acid oxidation, it seems like that could be like more detrimental because they don't have the fatty acid oxidation to pull from with stopping the glycolysis. Am I understanding that correctly or not
Yeah.
quite?
So the DCA, first of all, also too, you are stealing away. So for the kids with, it actually might be glucose supplementation actually might be more effective in the kids with fatty acid oxidation disorders, right? Because in the DCA treatment, glycolysis is still working. It's just that the carbons are being pulled into the citric acid cycle. I know this gets kind of heavy into biochemistry.
Yeah,
It's
okay.
not that you're inhibiting glycolysis, you're pulling away because the virus
Oh,
uses glycolysis, that very short pathway at the top to use its carbons to make nucleotides, to make fats, to make the things it needs to form a fully formed virus. You're pulling the kind of carbons away from that and putting them into the TCA cycle and eventually into the mitochondria. So they will
okay.
burn that in the mitochondria and make energy from that. So in
Okay.
that particular case, supplementation with, it might not work in our mice because their mitochondria are broken,
Hmm.
but fatty acid oxidation, it probably would work quite well because their mitochondria, I know there are some, there may be some inhibition there, but they still should be able to burn glucose, relatively well. And it is, a good way to help someone through an infection.
That's fascinating. Thank you so much for kind of breaking that down again. We had a patient ask, you mentioned a little bit that all of this can be applied to the generalized greater mitochondrial population. I'm
Yeah.
wondering if you could just take a second to explain maybe why that is or expound on
Yeah,
that a little bit.
absolutely. So the reason is because anytime you have mitochondrial dysfunction, whether it's from complex 1 deficiency, complex 4 deficiency, mitochondrial depletion, it all leads to decreased OXPHOS, right? The ability to generate ATP in the mitochondria. So in that scenario, regardless of what the genetic problem is, they will upregulate glycolysis, right? So our findings are actually applicable to many different mitochondrial diseases, even though we just studied a Leigh syndrome model specifically, because one of the shared things amongst many different mitochondrial diseases is the increased use of glycolysis, because that's the way they generate ATP. You can still generate ATP. It's less efficient than the mitochondria. So therefore, yeah, that's why they ramp it up, right? If they basically turn the hose, they turn the faucet fully open, because you can get more water that way. And that's because, the downstream doesn't work. So therefore, that's what they try and do. The cells try and upregulate glycolysis as much as possible to try and make up for that deficit from the mitochondria. So that's why our results are kind of applicable to, all different types of mitochondrial diseases because they all do very similar things.
The Pol G patient who asked, is there anything that we can do to boost the immunity in our cells? And when you talked about the cytokine
Yeah.
storms in particular, she said Is there anything that you think would help the inflammation that comes with the cytokine storms? Um, can you
Yeah.
speak on any
So
of that? I know it's kind of tricky, but
no,
I'm
I mean
curious what
there
your
so
thoughts.
there aren't really any treatments per se, but at least none that are being targeted specifically for this situation in mitochondrial disease, But in general, some of the so obviously vaccination I mentioned, hygiene I mentioned, but okay, so then what about when you actually get sick? Tamiflu, as I said, within the first 48 hours, that's kind of that critical period where it's the most effective. So if you feel like you're coming down with something, it is important to kind of get tested, pretty quickly, and use Tamiflu. And also, as I mentioned, Tamiflu is effective against influenzas A and B. It doesn't seem to be affected by the fact that flu changes every year. So it can be quite an effective medication. It doesn't prevent you from having flu. It decreases the number of days that you're sick. And for any person with mitochondrial disease, or even fatty acid oxidation disorders, or any type of inborn error metabolism, the number of days we can shave off of them being sick is just to their benefit, essentially. So what else can people do? It also has to do with kind of keeping a healthy diet, eating, your fruits and vegetables, which are rich in antioxidants, which of course are important for immune function as well as fighting off viruses and things like that. So while I can't really give like a very specific, kind of directed recommendation that we do, some of the things that you kind of already know and are encouraged to do are the things that I would recommend.
The same patient is asking, is DCA available to take? Or where
Yeah.
is that in the
So
process?
Yeah, so DCA is currently it's in trials actually, for pyruvate dehydrogenase deficiency, which can cause Leigh syndrome as well as a variety of mitochondrial disorders, which are different than Leigh syndrome. It can be quite mild or it can be quite severe, and the severe end is Leigh syndrome. So the reason is what they're trying to do is they're trying to decrease some of the lactate that builds up, right? So when you exclusively use glycolysis, one of the end products of glycolysis is unfortunately lactate, right? So some people in the audience may know that, sometimes your doctor will check lactate in your blood to kind of see how you're doing. If they have too much sugar, right, they can have a buildup of lactate in their blood. Individuals who don't have mitochondrial disease, they accumulate lactate by running marathons and things like that. So lactate's a normal process. It's just that because there's overuse of glycolysis in mitochondrial disease, they tend to have a lot more. Um, so um, so yeah. So getting back to the original question, can you just remind me? Did I answer it or did I go off on a tangent?
I think, yeah, no, I think you did because the original question was about DCA and if it's
Oh yeah,
available.
yeah,
And
sorry.
so,
So it is
yeah.
available. Yeah, so it's available. So it's in trials for pyruvate dehydrogenase deficiency. And that's Peter Stackpool at the University of Florida who's doing that. Who's a great individual. Um, it was tried years ago for mitochondrial disease, other types of mitochondrial disease due to other things besides PDH deficiency, and so it unfortunately in long-term treatment, it caused a peripheral neuropathy. So the side effect was actually considered, you know, very, very undesirable, in mitochondrial disease patients. However, short-term treatment, which is kind of what we're talking about here, may be a possibility in the future. I don't know. We're still kind of doing more studies, to determine, because it wouldn't be long-term, right? It would only be during the illness itself to try and kind of shut it down, or at least decrease the severity of the illness. So it wouldn't be long term. So those side effects wouldn't be expected essentially. So it's not currently available, and the only thing is it's in clinical trial for PDH deficiency.
Okay. No, that's super helpful. And it's
Sorry,
also
that was
helpful to know.
long-winded, but
No, no,
yeah.
no. It's really helpful to understand the implications, especially of peripheral neuropathy in our community, but the notion
Yes.
that maybe short term could be in our future at some point to help prevent illnesses carrying on for such a long period of time. Because I know a lot of people in our community really struggle with that, with the illness just going on and on. And then maybe there's a threshold where like it's gone on and on and on, and then they can try something like this to nip it in a bud once it gets out of hand.
So the other thing we have tried in these mice, which I didn't mention today, is we've done immunoglobulin therapy. And immunoglobulin therapy is equally effective in terms of squashing, the virus. Um, so that's mice, right? So in
Yeah.
human beings, immunoglobulin therapy has been shown to be effective. The studies that I'm thinking of were in the ICU, and these were in individuals who did not have mitochondrial disease, but it was during the 2009 H1N1 pandemic, where these individuals were really, really sick and in the ICU and kind of, really moribund, and they gave them IVIG, and they actually saw improvement essentially in those individuals. So that's another thing that we are, as a group, we are looking into, because there are a number of individuals in the mitochondrial disease community who are on immunoglobulin replacement therapy. And the anecdotal evidence that we are hearing is that they do much better. Actually, they're not in the hospital as often, and there's multiple reasons that that may be, but yeah, that's the other thing I just wanted to mention.
No, that's really helpful. So because I do know patients that have used IVIG and have had good experience, has there been uh, has there been a study, like an overarching study, or is it primarily case studies at this point that physicians are using in regards to
Yeah,
that?
so you mean in terms of like justifying being able to give it, or
Mm-hmm.
yeah, it's so the stories that we hear, it's kind of one of two reasons. One of which is the child gets diagnosed with an immunodeficiency, so they go to an immunologist, they fulfill all the criteria, and then they get put on immunoglobulin therapy. The other one that we've heard is that there are some providers where they kind of hit a brick wall in terms of the person's treatment, whether it's like controlling their seizures or something like that, and they say, let's just try it. And so it's kind of, it is anecdotal evidence, but they say like that, the patients are doing better, they're not in the hospital as often. They are even saying that it helps control their seizures. But this is all kind of word of mouth at this point. So we are actually putting together a study to study individuals on immunoglobulin replacement therapy, and hope to be able to provide that to patients in the future as part of our study.
Oh, that's amazing. That's amazing. I know we're past 1 o'clock. Do you have a couple more minutes? Because there's a
Yeah,
lot.
sure.
Okay. So you mentioned the seizures, and there is a patient
Yeah.
that brought up, is there a way to treat or prevent increased seizures for kids with mito who've been exposed to viruses outside of the regular seizure rescue meds? So you mentioned the IVIG. I don't know if there's anything else you'd want to say to that while we're on the topic of seizures.
Yeah, to be honest, we haven't really looked at that question yet. And that's something that we plan on doing, because we discovered that I mentioned a little bit earlier, kind of in a passing note, that our mouse, that when we give it flu, about 2 days after they get flu, they normally have seizures, but now their seizures like increase like dramatically. So I don't have an answer to that, but I want to reassure the audience that it is something that we're actively looking into, that can we kind of intervene during that period to help kind of reduce, the burden that they're experiencing, because, obviously seizures, there's a lot that comes with it, right? The seizure itself can be life-threatening. There can be issues with breathing, respiration, that the individual can stop breathing. Like there's a lot of things that go with seizures, which can make them dangerous.
Mhm.
While I don't have an answer, I can say that we are actively looking at that question, whether they're beyond just the normal kind of rescue meds that we have and things like that. Are there ways that we can intervene that actually have to do specifically with the infection itself?
No, that's very encouraging because sometimes it's just as a patient or a parent, just knowing that you guys are evaluating the questions that we have, like is so encouraging, even if the answers aren't there yet. It's really helpful.
Yeah, we
Um,
were, just to say, when we saw that in our mouse, we were elated. I mean, bad for the mouse, and we're not like celebrating, that, but it was great that this actually recapitulated something that our patients actually report. Like, so the
yeah.
paper we published a couple of years ago during COVID, looking at what people were exposed to, and then what viruses they were exposed to, and then what were the outcomes of those viral infections, increase in seizure rate was one of the most commonly reported things that we saw. So this is, and then of course we hear it from our patients as well. So it is obviously really seems it is happening in the community. So if there's a way that we can, decrease that risk and decrease the morbidity that results from that, it is something, as I said, it's a question we're actively pursuing.
Well, and I think too, just the your experiment and the process and being able to see the increase of viral load in the mouse's lungs, like to me,
Yeah,
that in and of itself is huge because it's affirmation as to what we see in our kids, right? We already
right.
know, like as parents or as patients, like, yeah, this virus seems to last forever, but
Yeah,
to be able to actually see it, it's just very affirming because sometimes people are like, oh no, it's just a little bit longer, but no, like it's so we really appreciate you doing that research because it provides a lot of understanding for us as well. Um, so a patient did ask if there is a Tamiflu that doesn't contain dairy that you know of.
I don't. I don't. I'm sorry. I've never been faced with that question before. So I apologize. I don't know whether there is.
Um, and then there have been questions that have come in about explaining the 2-DG. If you could explain a little bit more about that.
Yeah, sure. So that's a medication that not a medication, sorry, it's a chemical that you can use to block glycolysis, and it's used experimentally. It's not used, in patients. And so the way it works is that cells, when they initially take up glucose, it goes through a transporter through the cell membrane into the cell, and then glucose gets phosphorylated. In other words, it sticks a phosphorus group on it. And the point of that is that so it doesn't escape the cell, right? If you don't put a phosphate on it, then it can just go back same way it came in, essentially, right? So you want to keep it inside the cell. So you put a phosphate group on that. 2DG looks like glucose, right? So it comes in and it gets phosphorylated and then shuts the rest of the pathway down. So you're not shutting it down to zero, but you're basically kind of like lowering the flow through glycolysis. So you can use it in mice. It's not really being used in patients. Because I think because it is so widely, it acts in essentially every cell type. You can imagine that it does have a lot of toxicity. Yeah, that's what 2DG. It stands for 2-deoxyglucose.
So theoretically, DCA or 2DG, would these interventions be exclusive to influenza, or do you think there'd be this similar kind of effect with rhinovirus,
That's a
COVID?
great question. So even though I focus on influenza today, a lot of viruses use a similar strategy. Not to anthropomorphize viruses, like they're thinking about this, but like through evolution, how we have co-evolved with viruses. Um, a lot of them use the same technique where glycolysis is upregulated because it helps make a lot of stuff, which it helps make the Legos to build another virus, essentially, right? So it could potentially be effective against other types of viruses as well.
All right, I'm gonna let this be the last one, and I'm sorry, there's so many questions, and
No,
they're
no.
all really, really great. Um, this one is in regards to Elhan in particular, and it's a multifaceted question. So I'm just gonna read it, and I know it's a lot, but I want to make sure that I capture the whole thing. It says many people affected by Elhan report that they feel they get sick more frequently than others, and that it takes longer to shake an illness. Since Elhan is a complex 1 mitochondrial disease, is it reasonable to think that there could be a correlation? Or is it more likely that when someone is affected by LHAN, they're simply more aware of illness and are seeing casualty when it's not really there? Or do we just not have enough information about LHAN and viruses to have a reasonable hypothesis?
So the answer is C. I don't know if we have enough information at this point. I mean, I haven't really seen many studies that really looked at viral infection in individuals with Alhan. However, complex one deficiency is the complex one deficiency that they have. Is we were just looking at complex one deficiency now. We haven't looked at other complexes to see whether they behave the same way. But we think they would, because we think the central issue is that glycolysis being upregulated. And to be honest, it doesn't matter which complex you inhibit, the
Okay.
response is still the same. Um, so there isn't a lot of work on LHON, but I would imagine that some of the things that we found with regards to immune dysfunction in kids with mitochondrial disease with different types of complex deficiencies, as well as some of the data I showed you today with, the increased susceptibility to infection. I would imagine that a lot of it would also extend to LHON as well, but I don't know of anyone who's formally kind of looked at that.
Okay. Thank you so much, Dr. McGuire. I just want you to know how much the community just appreciates you and appreciates all the hard work that you're doing at the mini study and all the things that you're learning. We really do feel the effects of that, and we know how much you care about this patient community, and we really, really appreciate that. And we always appreciate you coming on and explaining more about what's going on with your research and where things are at. It just helps us to understand more, and it gives us hope. You bring us hope. So thank you so much for coming on today. We appreciate it. And thank you for everyone who has joined us this afternoon, to the patients and the families and clinicians that have taken time to join us on the call today. And for those that are listening afterwards, I know several people have asked if this would be recorded because they wanted to make sure that they saw it afterwards or to be able to see it twice. So thank you for joining us and asking your questions. Together we learn. I hope that you'll consider joining us next month with Dr. Alessandro Pregoni, who will discuss modeling mitochondrial disease with patient-derived stem cells and brain organoids. So until next time.
Thank you very much.
Thanks.